So welcome everyone to today's Wednesday afternoon lecture. I'm Susan Gottesman. I'm here on behalf of the Lambda Lunch Interest Group. For those of you who don't know who we are, but I think this audience mostly does. We're a weekly seminar series on primarily prokaryotic topics, but other interesting science as well. And we invite you to join us, and we every year nominate our favorite people who haven't been here to come and give walls, and we're fortunate enough to have Nancy Moran selected from that nomination for this year's Walls talk. So Nancy is the Warren and Viola May Raymer Chair Professor at the University of Texas in Austin. She's in the Department of Integrative Biology, and I think most of us who have been in this field a while recognize that some of the most interesting microbiology and just interesting ideas in general come when we sort of have people from very different viewpoints come together and bring their science to what we're doing. So I think that Nancy is a wonderful example of that. She got her undergraduate education at UT Austin and her Ph.D. at Michigan in zoology. And she, I think, got interested in insects, aphids, fairly early in her career, and then incorporated the microbiome in about 1990, and really has been doing groundbreaking work on the intersection of the microbiome and insects. She was recognized with the MacArthur Fellowship in 1997, election to the National Academy of Sciences in 2004, a whole long list of prizes, including a lot from molecular ecology, which is not something that us microbiologists are normally thinking about, and in 2023 received the NAS Selman-Waxman Award in microbiology. So after her PhD, et cetera, she established her lab at the University of Arizona, where she did much of this work, moved in 2010 to Yale for a few years, and then moved from Yale to UT Austin, where she's been from 2013 on. So we're delighted she could come today. Now, if at the end you want more, please join us tomorrow for Lambda Lunch at 11 in Building 37 on the 6th floor. And if you don't know where to go or anything, just catch me at the reception. And there is a reception after this. Other important information. The CME code is on the board. It's 50096. If you are online and want to ask questions, you can send live feedback at any time. Use the send live feedback button at any time during the talk, or certainly at the end. For those of you in the room, I guess, unlike Lambda Lunch, we'll save the questions for the end, but at that point we'll ask that you come to the microphones so that everyone, including those online, can hear it. So I'm delighted to have you here, Nancy. Thanks. This is her first visit to NIH, which tells you this is long overdue. So thank you for coming, and we're looking forward to the talk. Thanks.
All right, thanks, Susan, and thanks to everyone else who's been organizing this really fun visit. I've had a lot of great talks with people here today, and I'm looking forward to some more for the rest of today and tomorrow. So I'm glad to be here and learning a lot of new things. Today I'm going to give a very broad talk to kind of cover this very broad field that's developed a lot and that I've been part of really since the field started around 1990. And basically it's exploring how microbes interact with insects. And I've always loved insects. I'm one of those people from a little kid stage that loved insects. But it's been really a revelation to realize that a lot of what we see in insect biology is due to the microbes living in them, specifically bacteria in most cases living in them. So I'll kind of give a few of the most exciting examples, I think, to show the different ways that this happens. So insects, okay, you might not know, but they are more than half of all species that are described of everything. They are very old. They originated really when life came onto land. I mean, they're very ancient, along with the first plants on land about 450 million years ago. And they have these symbioses, but they were rarely studied until about 30 years ago when molecular tools became available for looking at them. And then there's been an incredible increase, especially since 2007. And you might recognize that year as the year when next-gen sequencing became widely available and cheaper, and it really led to a lot more studies. And there's a huge variety of these interactions. So I'll give you some examples of the different groups. I can roughly put them into several categories. And the first one I'll talk about are what we sometimes call primary symbionts. These are obligate for the host. The host cannot develop without them. They're strictly maternally transmitted from mother to daughter, from mother to son too, but he's a dead end, only from mothers. So just like mitochondria. The genomes are really characteristic, and I'll talk more about that. They're highly reduced. They're very, very, very extreme genomes. And examples are a couple of these, and I'll talk more about the examples, especially the Buchnera and aphids, which has been the central model. So these occur a lot in insects. They are not restricted to insects. They're actually in a lot of other invertebrates or even in some fungi. But most of the cases that have been studied are in insects. And then there's the so-called secondary symbionts. And these are ones that, again, are mostly transmitted maternally. So in the lab, you can maintain them for hundreds of generations. They'll be strictly maternally transmitted with high fidelity. But we can see that in nature, they're jumping around sometimes. They're horizontally transmitted in nature. We can see that by doing phylogenies and so on. So we know, and they're more dynamic in terms of their genomes. They have recombination and bacteriophage and things which are not present in the primary symbionts. A really common one of these is Wolbachia that is in insects. It's jumping around, and most insect species have had it in their history, probably all. And Hamiltonella is an example I'll talk about in aphids. And then we come to things that are more like gut symbiont, that are more like gut symbionts and often are gut symbionts. And this category where there's host-specialized symbionts, they live only in the host. They don't replicate in other environments. They're restricted to the host, but they're not maternally transmitted. They're just socially transmitted, just like our gut microbiota. And then finally, just environmental microbes that might colonize hosts. They might have a big effect on the host, but the microbes themselves live in many different places. So they haven't evolved specifically with hosts. So first I'll talk about these so-called primary symbionts. These are inherited. They're intracellular. Usually, there's actually some cases where they're not, but mostly they're intracellular. And they generally live within specialized host cells. And these have evolved again and again. They're called bacteriocytes, so they're cells of the host that house the symbionts, usually in the cytoplasm, and often the cytoplasm is basically just stuffed with these bacterial symbionts. They've evolved over and over, bacteriocytes, from different cell types. And sometimes in the same insect, you can have two different cell types that house different bacterial symbionts. And this is just one example. The example I'll talk most about, here's an aphid, and it has its gut. And then these yellow things are the ovarioles. And the bacteriocytes are in the body cavity here. And if you just look at one of these tiny embryos, so aphids have prenatal development and the embryo is within the mother. This is the embryo at a very early stage. And basically almost the entire embryo is the symbionts here. So basically they're a very big part of development, in many cases early embryonic development. And this is just showing the later stages, but one important point is in most cases they are actually enclosed in a host-derived membrane. So they're in a vacuole within these bacteriocyte cells. Well, they're so dominant in embryos that early embryologists were looking at insect embryos and other invertebrate embryos, trying to understand development and using light microscopy. And a very prominent one who noticed these symbionts was named Paul Buchner. He was an embryologist in Germany. He documented hundreds of animal symbioses, including hundreds in insects. He had a lot of treatment of aphids and their symbionts. And this was all based on microscopy. And he came up with some ideas. One, he believed that these symbionts, this type of symbiont, was providing nutrients to the host. He believed that they were ancient in these hosts that evolved with them a long time. And he believed that the host was in control, that these were kind of passive things at this point, and that the host was in control of the symbionts. He sort of argued this. He published, his work was translated into English in 1965 in a big book that has all of this kind of ideas in it. It has a lot of these were mostly right, these ideas, but he had a lot of wrong ideas also in that book because obviously he couldn't know a lot about what was going on. So what have we learned since then? So I'll be using this aphid Buchnera, named after Paul Buchner, symbiosis as a kind of central model. It really probably is the most studied model of this. And so in 1989, Paul Bellman at UC Davis was the first to characterize an unculturable symbiont using DNA sequencing. He used PCR, which was relatively new at the time, and then actually cloned and sequenced the 16S ribosomal RNA. He then gave it this bacterium a name, and he noticed that it's close to E. coli. It's in the same group of gamma proteobacteria. So then luckily, he called me up one day and said, "Do you want to work on this?" Because I was working on aphids, and I started working with him and dropped the other things I was doing, and ever since then have primarily worked on symbionts. In 1993, we did some work looking at molecular phylogenies for these symbionts and comparing those to phylogenies of the host. And it turns out that the host and symbiont phylogeny map onto each other exactly. So in other words, they've evolved together, and when speciation occurs in the host, you get divergence in the corresponding symbiont, which is what you'd expect if it's strictly maternal. But the time scale is what's impressive because these are very old groups. So aphids based on fossils are at least 150 million years. So it dates back before that common ancestor. And then in 2000, the first symbiont genome was sequenced. This was early in genome sequencing days, and by Shigenobu et al. In Japan, and they sequenced Buchnera of P. aphid. And since that time, there are more than 12,000 papers on Buchnera in aphids alone. So there's really been a lot of work on this system, but as you'll see, there's still a lot we really don't know. So the problem I should mention about these symbionts is they can't be cultured in the lab, and that is why we knew so little about them. So Buchnera, we got a genome, and what was it like? Well, it turned out to be tiny. It was 580 encoded proteins, about 600 kilobases. It looks just, surprisingly at the time at least, it looks just like a reduced E. coli genome, basically a subset of E. coli genes. There were only two sort of novel orphan genes, and that's remained to be the case with many more Buchnera genomes sequenced. So many gene functions were lost. Most regulatory genes, most transporters, most DNA repair genes, and also many genes involved in making the cell envelope, peptidoglycan, LPS, and phospholipids. Many of those were missing as well. What was retained, it still retains all the genes from making it for translation, so making a ribosome, tRNA synthetases, DNA polymerase, so replication, transcription, and translation. Basic machinery for that is retained. So it's still like a cell in many ways. And then the other thing it retains, kind of impressively given how small these genomes are some biosynthetic pathways that are basically what is needed by the host. So Buchner had hypothesized they're making nutrients for the host. That turns out to be true. And in the case of Buchnera, it's making amino acids, essential amino acids, so the ones that are essential for animals broadly, including us, but also including insects. And on the right here, so aphids feed on phloem sap. It's basically a sterile diet that has a lot of sugar in it, and it has free amino acids, but the amino acid profile is very unbalanced from the point of view of animal nutrition. It's got all these non-essential amino acids, kind of the cheap amino acids, and then essential amino acids, which tend to have the longer pathways and require more ATP to make them. Those are the ones that animals in general have lost, and those are the exact set of pathways that Buchnera has kept, so complementary to what the aphid could do. And then some other work by Paul Bauman's lab back then, and also a lab in Spain, Andres Moya, showed that the Buchnera genome also includes some plasmids, and those plasmids are devoted to making extra copies of some of the genes that limit the rate of production of tryptophan and leucine, two of the essential amino acids. So it seems like specific adaptations to better produce for the host benefit. And So since that time, many of these nutrient provisioning symbionts have been found. People have done phylogenetics, our lab and other labs, to show that these are ancient in the hosts. And a lot of features, major features in the lifestyles of insects are due to these symbionts. So example, if you wonder why beetles sometimes are very hard, they have that hard cuticle, they have a symbiont that makes extra tyrosine, which is a substrate for making melanin, which is what makes beetles hard. And then cockroaches, they live on nitrogen-poor diets, and their symbiont recycles the cockroach's own nitrogen waste, the uric acid, and then also makes amino acids. And then whiteflies are sort of like aphids. They need amino acids essential amino acids because they feed on phloem sap, but in this case they also make carotenoids for whiteflies. And because I'm at NIH, I want to mention something relevant to health. These kind of symbionts also occur in a lot of insects important to human health, because insects that feed on blood generally are not getting B vitamins that are required in insect metabolism. So bed bugs, they have a special version of Wolbachia that makes B vitamins, so these lovely things. Body lice have another type of symbiont from the gamma proteobacteria that again makes B vitamins, but otherwise has this reduced genome and all these features that are similar to what I described for Buchnera. And then tsetse flies have a symbiont called Wigglesworthia, and it also makes B vitamins required for the host. So these are all blood-feeding insects. Well, so in some groups, it gets really complicated, and Buchner had a favorite group, which he called the fairyland of symbiosis. And it's a group of plant sap-feeding insects, and it's the Ochinarhyncha, it's called the clade. And they feed, there's a huge number. There are leafhoppers, cicadas, spittlebugs, all these plant hoppers. So all of these, you've all seen them a lot, and they're major agricultural pests in some cases, and there's a lot of species of them. Well, it turns out that they can have up to 3 or 4 different symbionts in the same insect living in different types of bacteriocytes. And very commonly they have 2. And it turns out with lots of work by some of the people in my lab, including John and Gordon shown here, that working out phylogenies and where these bacteria live and looking at, we can see that they colonize, a couple of bacteria were in the common ancestor of this whole group, about 260 million years, and then diversified with them. But in some cases, one of them would be lost and replaced by yet a different bacterium that sort of moves into the system and occupies a new cell type, a new type of bacteriocyte. So there's a bit of turnover, but there's sort of this tapestry of symbionts coming and going. So this really would have been a dream of Buchner to work out this kind of history. He actually kind of speculated about these kind of events. And what we can do that Buchner couldn't, sequencing genomes of these, it turns out that when you look at a pair of these symbionts that are in the same host, they have exactly complementary nutritional capabilities, so especially in terms of amino acids, which is the main thing they make. So you'll have one making 8 of the essential amino acids, the other one making 2, or you'll have one making 7, the other one making 3. And they pair up, and basically the redundant pathways that must have been present when they first entered the symbiosis, when they all had all the pathways, get lost, and so you pare it down to the host has these pathways, each symbiont has a complementary set. So it's really fit together very beautifully and really kind of gave you confidence in using genomics to understand what's going on. So one thing that's unexpected in all of this that I mentioned briefly is these are tiny genomes. In fact, the tiniest known cellular genomes are in these insect symbionts. The record is 120 kb, I think it's 130 protein coding genes. It's the bare minimum for making a ribosome, translation, transcription, replication, and making a few nutrients for the host. And so we keep discovering these. They have evolved over and over. This is just showing a graph of genome size and number of protein coding genes. In bacteria, it's really nice, 1 kb, 1 gene on average. But the tiny insect symbiont genomes are down here. So very, very small. Buchnera itself was not at all the smallest. Many smaller ones were found later. We found a number of those in different insects. For a little while, all the smallest genomes of cellular organisms were from Tucson, Arizona, where my lab was, because we just go out and find them. It was nothing about Tucson. They're all over the world, but it was just a matter of discovering them and then figuring out, using the benefit of next-gen sequencing, that these things were out there. So how do these things work? And the host has evolved a lot to control and support these symbionts. We can tell that. What we don't know is the real mechanisms, and just the experimental limitations of these systems are really great because we can't grow the symbionts separately from the host. But using genomics and transcriptomics and things, we have done a lot of work trying to look at what genes are expressed in the bacteriocytes, how do those change during the life cycle of the bacteriocyte and of the symbiosis within an individual host lifetime, and how do those work together. And I won't go into details, but just mention that there are these very clear coordinated shifts in what genes are being expressed in the symbiont and in the host, in the bacteriocyte in which it resides. And so we have shifts in metabolism to different states, and it seems to be coordinated and predictable through the life cycle of the aphid. So how this happens, what molecules are going back and forth, there's a lot that we don't know about it. And so this is, over here, this is from Julie Perot's work showing what I showed earlier. Here's an embryo of an aphid, and this green here is the Buchnera. And actually most of these outer cells are actually the maternal follicle. The embryo only has relatively few cells at this point. So basically it's been worked out that there's a specific stage in embryogenesis where a large syncytial cell is exposed to the body cavity of the mother, and a nearby bacteriocyte somehow senses that. We don't know what the signal is, and exocytoses Buchnera just free into the body cavity. They don't have flagella or anything, but some of them contact this syncytial cell and are taken in, endocytosed, and then later are packaged into bacteriocytes. And then some work by Simonet et al., a lab in France, has shown that actually the bacteriocyte has a very distinctive life cycle, and they start getting killed off later in the life of the aphid in a very precise way that involves kind of the proliferation of the ER and vesicularization of the whole cell and the symbionts dying. And so that happens through the later life of the aphid in a predictable way. Okay, another thing that was really unexpected is that it turns out that in the bacteriocytes, some of the highly expressed genes that are only expressed in bacteriocytes and not in other cell types of the host actually are bacterial genes that were ancestrally transferred into the insect genome. We keep finding more and more cases of this, so it's usually not a large set of genes, but they're highly expressed in these bacteriocytes. And originally when we got the aphid genome sequenced, it was sort of one of the last of the Sanger sequence animal genomes published in 2010. Nakabachi led this work. Has his own lab in Japan now. But he basically looked into these horizontally transferred genes, how they're expressed in bacteriocytes, did a lot of phylogenetics, and showed where they arose. So here's a gene, an aphid gene for this gene RLPA, and it's most related to these other things which are bacteria. These are alpha proteobacteria, and its closest relatives are this group Wolbachia, which those are insect symbionts. So these bacterial genes didn't come from Buchnera, the resident symbiont of aphids. They came from a different bacterium, Wolbachia, which is a bacterium that often lives in the germline cells of the insect. So Buchnera is primarily only in somatic cells, the bacteriocytes. It's very difficult for a gene to jump from a somatic cell into the germline of the host. There's quite a few membranes in between, and probably much more frequent that the opportunity arises if the symbiont is actually in a germline cell. So that might be a reason that we've repeatedly found in different systems that the source of these horizontally transferred genes is often Wolbachia or in the Wolbachia cluster. Nakabachi went on in his own lab to show that the protein product of one of these genes, RLPA, it's involved in peptidoglycan production and recycling of peptidoglycan subunits, is basically expressed only in bacteriocytes, and he showed that it does actually enter the bacterial cell using antibodies to it and so on. So here's some EM with gold particles where he could show it's there. So we know it's actually pretty rare that we know anything about molecules going back and forth between host and bacterium in these systems, but this is one case where he showed it. And further work by Tom Smith in my lab and showed that some of these horizontally transferred enzymes, he took the versions that are in aphids, put the transcript into E. coli and looked at the activity and did a lot of biochemistry and showed that they have similar activities to what they would do in E. coli. This is in recycling pieces of peptidoglycan. They're similar, but actually have some additional reactions that would be minor reactions for the E. coli versions, but they've shifted activity a bit. So some genes are transferred to the insect host, probably shifted their activities to some promiscuous reaction that was minor in the past, and now are serving some purpose in recycling peptidoglycan in this symbiont. So somehow the host is acquiring the ability to control and produce the cell envelope of the symbiont. So Buchnera is just one case. Another case that was shown earlier by John McCutcheon's lab, in his own lab, he was in another group of insects called mealybugs. They also feed on plant sap. They're scale insects. And they actually have some of the peptidoglycan biosynthesis genes that are part of the insect genome. So there's kind of these mosaic pathways where some of the genes are produced by the bacterium, some by the insect, and when you put them together, they fit to make the whole pathway. So experimentally, it's very hard to work on these, but it's very striking that you have this kind of complementary contributions from host versus symbiont, and he can show that they're expressed in there. So besides just losing genes, these obligate symbionts, they also, even the genes they retain undergo this kind of rapid and it looks like deleterious evolution. So the proteins themselves are evolving super fast, and that's shown by this phylogeny. So this is a phylogeny representing all bacteria, or, numerous phyla of bacteria. So the common ancestor is the ancestor of all bacteria. And the different major phyla are in different colors. And all of these extremely long branches, so this is a protein-based phylogeny based on amino acid sequences, these extremely long branches are all insect symbionts. So basically, they have genes homologous to those of other bacteria, but they're going crazy in terms of the rate of evolution. And you can show that the produced proteins are thermally very unstable. All of these symbionts convergently have evolved very high expression of heat shock proteins. So GRO-EL, DNA-K, or chaperonin is GRO-EL and DNA-K, are very highly expressed. Like it'll be 10% of the protein in the bacteria in the case of Buchnera. And you can't really express them in E. coli, say, without overexpressing chaperonin. So basically, they're very unstable, and the symbionts have evolved to kind of prop them up constantly by having these high levels of heat shock protein, which is expensive. And in fact, the symbionts seem to be a major reason for the thermal limitations on the host. So the insect ability to withstand heat is limited by the symbionts, it seems, in many cases. So in the case of Buchnera, if we give it 4 hours of heat on its second day of life, to the aphid, you'll get a drop in the levels of Buchnera over time following that, and you get lower survival. So survival relative to a non-heat shock control just drops dramatically along with the drop in Buchnera titer. And we have some cases, we have a little bit of experimental capability, figuring out a way to replace the resident Buchnera with a different Buchnera from a different matriline, and that way we can have a common host genotype, the same host genotype, with two different Buchnera. And we can put in a more heat-sensitive Buchnera, a more heat-tolerant Buchnera to replace a heat-sensitive one, and then we can basically rescue the aphid fitness so that it survives better following heat. So the heat sensitivity is due to symbionts, at least in some cases. So people in symbiosis field always want to invent new words. I don't know what it is. It's kind of a field that's grown a lot, but you hear these words. So some people say, well, they're just organelles. Are they just part of it's a meta-organism, or it's a hologenome? And I don't mind the different words, just so you know what reality you're actually talking about. And here's this quote from Joshua Lederberg in the paper where he coined the word plasmid. But how shall we choose the boundaries of the gene complex that constitutes an individual organism? Well, there's no easy solution because you have all these different combinations of shared fitness interests over time. And sometimes new words such as plasmid are very useful. Sometimes they're just very confusing. But in the case of these symbionts, they're actually not just organelles. I mean, at least they're quite different from mitochondria and plastids in that they retain most of the cellular machinery for replicating themselves. And they have very few proteins that are needed for that basic machinery. They only in the cell envelope kind of arena are host-encoded proteins apparently important. And they are extracellular a little bit during transmission to progeny, so part of their life is extracellular. So they're different, but they do have a lot of similarities. So then I want to move on and talk about one step away. So these secondary symbionts, they're transmitted maternally, but sometimes they're jumping horizontally, and we can see that by just looking at their distributions among insects in the field. A very famous one, probably the most widespread of all symbionts, is Wolbachia. It's in basically all insects have probably had it in their history. Many current species still have it. And the one but I haven't worked on it. One I'll talk about more is called Hamiltonella, and then we'll just I'll tell you about it. One we're working on right now, just a little bit. But actually, I was going to say that sometimes this first category, we call this the symbiotic rabbit hole. It's basically once the symbionts go down this, they're just getting smaller and smaller and more decayed, probably becoming kind of a weakness for the host because they're limiting its thermal tolerance. The host is now dependent on them. Its whole development has evolved around the presence of these symbionts. So it's very hard for them to just lose these symbionts. But this is something that we've seen. It's such a striking genomic syndrome in many different origins of it in different insect groups. So I'll talk about these secondary symbionts, especially this one, Hamiltonella defensa. It's a favorite of mine. It's non-cultivable. You can sort of grow it briefly with some insect cell lines. It's mostly maternally transmitted, occasionally horizontally. It's not obligate for the host. The host can take it or leave it. You can cure the host of Hamiltonella, and they'll live fine. And they're a little bit more pathogen-like. They're actually entering new hosts that don't have machinery to take care of them. They're able to enter a new host and to be vertically transmitted and persist there. So they live in the hemolymph, but also in a few bacteriocytes. So here, green is Buchnera, the red is Hamiltonella. And they're sort of rod-shaped, but they're very large cells. I didn't mention that often the obligate symbionts like Buchnera have very large, sometimes round, sometimes just pleomorphic branching cells. They usually have very, very large cells, even despite having very, very tiny genomes. So aphids have a lot of enemies, and one of their main enemies are these little wasps that lay an egg in the aphid. The egg hatches, a larva comes out, it selectively feeds on the aphid tissues, and then finally in the end it kills the aphid, and the aphid turns into a little cocoon and a wasp comes out. And so that's what happens if the wasp is successful. But it was observed some time ago by people just looking at susceptibility to these wasps in natural populations of pea aphids, that there's a huge variation. So if you expose them to the wasp, some of them will be totally fine. No aphid will die, no wasp will come out. Others, pretty much all the aphids will turn into wasps. So there's this huge variation in susceptibility. And this was just assumed to represent genetic variation in the aphids. I should mention one reason they could do these experiments easily is that aphids are clonal, so you can have clonal lines and grow them and do tests on them in the lab and have constant genetics and compare them. So this was actually of interest even just for biocontrol, because these wasps are used in biocontrol of aphids. Sometimes they work, sometimes they don't work at all, and it's due to this kind of variation. So it turns out, to make a long story short, but then I'll show you a little about how it works, that this variation results from different Hamiltonella being present. And so we can take in the lab the same aphid genotype, and we can have it with and without Hamiltonella. By microinjection, we can establish it 100% maternal transmission for very long periods of time. I've had some of them like 20 years. And so it's very stable, maternally transmitted, and you can compare with the background genotype being the same. And that's, in this graph down here, that's what's here. So here's one with no Hamiltonella. Most of them get parasitized. Here's one with one Hamiltonella. Again, most are parasitic. But then these two are highly resistant. And so you wonder why. And it turns out that these bacteria have bacteriophage. This one's called APSC, a sort of Siphonpisum secondary endosymbiont phage. And it turns out that when this phage was present in these earlier experiments done, like Kerry Oliver was a leader, he was in the lab and did some beautiful experiments to work all of this out. Turns out that the ones that are resistant have this bacteriophage. And then when you look at the bacteriophage genome, this was work done by Patrick Degnan. There's basically the bacteriophage have very similar genome, very little difference, except for a cassette within it that is totally different in different varieties of these APSC. And this little cassette encodes different toxin genes. And it turns out these were actually known toxins from other bacteria and from pathogens. So Shiga toxin-like, at least homology to Shiga toxin, cytolethal distending toxin, and these YD repeat toxins. So different ones of these phage have different ones of these. It's just hypervariable. And if you look in the field and collect aphid clones, so each of these columns is over here is from the field and over here are experimental. Basically, the ability to resist the wasps is just totally correlated with having the APSC. If you have Hamiltonella alone, it's as good as nothing. It's not having Hamiltonella at all. So basically, it is very predictive in terms of predicting susceptibility. And then in the lab, sometimes you get spontaneous loss of APSC because it recombines out of its site on the chromosome. And so you can pull out lines that don't have it. And it's actually somewhat, if there's no parasitoids around, it actually lowers aphid fitness a bit. So it's basically not adaptive to Hamiltonella to have these phage, which cause it, the cells to explode, unless there's parasitoid pressure. So in the lab, you sometimes lose it, and we could show that basically it's loss corresponding with total loss of this resistance. And this one other version, this is some current work done by Jerry Maeda, current grad student in the lab, on a different symbiont in pea aphids called Fucatsuia symbiotica. And Jerry just very recently has successfully cultured this thing, so we're really excited because it's the first one of these symbionts you can actually culture. It's very slow growing, but it's a pure culture that persists. This has a very different way of protecting aphids. And so it was known a bit before, and Jerry's got more experiments showing that it gives resistance to certain fungal pathogens. So aphids not only have the wasp to worry about, but also fungal pathogens are major in different aphid populations. And it gives some resistance to different fungal pathogens. And he could show that if you just take an extract of infected versus uninfected aphids, that it basically inhibits the growth of fungi of different fungal types on plates. But at the same time, it's very costly. It's kind of a puzzle that something vertically transmitted could be this harmful to its host, because there should be selection to help the host. It causes these deformed embryos, so normal embryo, weird deformed ones that have eyes even when they're still very tiny. Just all the timing of everything is messed up. So it causes these deformed embryos and very low fecundity that's worse at higher temperatures. And so he's done some work figuring this out, and it seems to all boil down to this one large locus in Fucatsuia, this a polyketide synthase cluster that's highly expressed, especially at the higher temperatures, which 20 is the high temperature here, so it's not that hot. It turns out this is a horizontally transmitted cluster from plant-associated, plant pathogenic bacteria like Pantoia. And some people in China had worked on the plant pathogens and just recently did some work and determined the structure of this polyketide molecule called erbicolin A, and had shown that it's antifungal and it can be used perhaps in controlling plant pathogens. And its target is this sterol that's in fungal cell walls, or gosterol. And so basically, in this paper, they did this work showing the structure and the likely target. And it turns out that insects have a sterol called ecdysone for shorthand, basically the insect molting hormone. It's very, very similar in structure to the sterol in fungal cell walls. So one hypothesis Jerry has is that this antifungal polyketide is great. It protects against fungi, but it messes up development. There's this kind of, two sides of it, plus and minus, from the point of view of aphids. So maybe it's interfering with endocrine signaling. Now he's doing experiments to try to figure that out. But he's recently isolated it, and they purified the compound and are working on the structure. So it's really nice that you can make finally some progress with a culturable example of this kind of symbiont. And I wanted to say a little bit about Wolbachia, which I have not worked on, but a lot of people have. And for a very long time, so it's maternally transmitted, can't be cultured. It was studied because it manipulates host reproduction, basically gives an advantage to the matriline's that have Wolbachia. They sterilize, basically males that have Wolbachia will sterilize the non-Wolbachia matriline's, which increases the Wolbachia bearers in the population. That was studied a lot. It was thought to be the main way Wolbachia spread. But then two labs separately, one in England, one in Australia, found around 2008 that in fact it gives broad protection against RNA viruses. This was first noticed in Drosophila. So mostly in most insects, people don't know what the viruses are. They haven't studied them. They're kind of an unknown thing. But in Drosophila is one of the few examples where we do know what the viruses are of these insects. And in some strains of Wolbachia give very high protection. So here the black line is survival of ones with no Wolbachia. Some of the Wolbachia-bearing lines would have a big advantage. And I mention it because it is, again, a little bit related to health, because now this was realized by Scott O'Neill. He was on one of the papers where it was first found that there was this antiviral effect. And he went into the idea of putting it into mosquitoes so that it would be an antiviral in mosquitoes, and specifically Aedes aegypti, the main vector of dengue. And so the idea here is that you have to get it into that mosquito first, and apparently that was very hard, but they finally did. And then you take those mosquito lines and you release them in areas where dengue is a problem and where those mosquitoes are living. And then it has, well, first of all, this reproductive manipulation effect can cause it to spread in the population, because the Wolbachia bearers can basically replace the non-Wolbachia bearers at certain frequencies. So you can write models and figure out whether this will work at a given frequency of release. And then the second effect is this anti-RNA viral effect. And then the idea is that you'd still have mosquitoes, they'd still be biting you, but they would not transmit dengue so well. And it seems like it's worked in some places, so it's a huge program, and I can't summarize it all. I just want to mention it because it's one place where insect symbionts have really started to potentially play a role in terms of human disease. They've now deployed this system. First they did tests in Australia, and then have spread where they've got sites around the world. This is just one random reference if you want to look up something about it. Or World Mosquito Program, they have a website. Okay, so now I want to move to the last categories, which I'll spend less time on, but mostly this one, host specialized bacteria, but they're not maternally transmitted. These guys have normal genomes, horizontal gene transfer, recombination, so they're nothing like these vertically transmitted symbionts in terms of what the genomes look like. So these are largely gut bacteria. They could be in other host locations as well. And for the last decade or so, this has been really the main system I worked on, and I'll talk more about it tomorrow at the Lambda lunch. But we've especially looked in the honeybee, and it has a very specialized microbiome. And we've concentrated on this community that's in the ileum, this one part of the hindgut, and specifically where this part of the hindgut is dominated by two species, Snodgrassella, which lives right on the ileum wall, and Guliamella apicola, which kind of layered on top of it in clusters. So these are found only in social bees. They've evolved with bees based on a lot of different work for over 80 million years based on the age of these bees. They're socially transmitted in the hive, but it's not strictly maternal. It's just going among individuals just like our gut microbes do. And the nice thing here is they're all culturable, so finally can do different kinds of experiments, and we're developing genetic tools which are getting more streamlined and more possibilities just by the week almost. So they acquire this just after emerging from the pupal stage in the hive, and they get them from their sisters, and the full community is established by about day 4. And that gives us this advantage that if we take the pupae before they meet their sisters and let them emerge in a sterile environment, they will be microbiota free. So they won't have any of the microbes that are the characteristic ones that take over the microbiota in the gut. And so normally these specialized microbes will be 95 to 99% of what's in the bee or in the gut of the bee. We can basically prevent those from establishing, and then we can do experiments where we can control what bacteria are in the bee. And so this is one naturally exposed, showing Snodgrassella in purple. This is one exposed to a single inoculation in the lab after 4 days, and you see very robust colonization. So we know a lot about what these do. And one thing they do, especially this one Guliamella, is break down recalcitrant polysaccharides that are in the pollen husks. So the bee has already extracted the good nutrients, the sugar, the protein, but these pollen husks that have plant cell wall components and all kinds of complicated molecules, Guliamella has collected genes, and all the strains differ, it's very diverse, for like pectinases and different glycosyl hydrolases and so on, for breaking down these molecules and using the sugars, fermenting the sugars that come out. And possibly, well, we know that at least some of the resulting short-chain fatty acids are absorbed by the host, because you can look in the hemolymph, the blood of the bee, and find them there. So there's some nutritional role. We don't know how important it is quantitatively. One thing we started to get into is it turns out that there's a lot of strain variation. I thought this would be a simple system. There's your 5 species, but each of those species, of course, has all these strains, just like in the human gut. And it turns out in Guliamella, sometimes those strains are really bona fide species. So Guliamella apicola and apis, they're both very closely related, but one of them, Guliamella apis, lives in this forward part of the gut, right where the nitrogen waste is coming into the bee, and it's able to use urea and basically ammonia for making amino acids, whereas Guliamella apicola cannot use urea, but it's got the specialization on all of these glycosyl hydrolases and pectinases and so on. They also have different pH optima, and we've measured the pH is higher in this forward part and lower back here where this fermentation is going on. So they seem to have diverged in their ecology within a single host. So every bee has both of these, every honeybee worker, but they're having this different metabolism and different niches within the gut. And Snodgrassella colonizes along the whole gut. Okay, but the main thing that bacteria do in the bee gut is protect against pathogens. At least that's what I think is the main thing. There have been a lot of studies from us and other labs showing that they protect against opportunistic bacteria, trypanosomatids, which are bee pathogens, and different RNA viruses that are known bee pathogens. It has some implications even just in beekeeping, because we've done work, and others also, showing that common beekeeping practices, such as giving antibiotics to the bee colony, or just agricultural practices such as herbicides that target pathways that are present in these bacteria, they disrupt the microbiota. They cause the in-hive mortality to be higher. So we can do mark recapture in the hives and look at mortality in their natural environment, because bees come back home. That's where they live. And so we can see how long they persist. And in lab challenges, we can show that disrupting with these compounds will cause them to be more susceptible to pathogens. So the microbiota is probably most important in protecting against pathogens. And this work by Margaret Steele shows that even, so these are monoinoculations by different strains, and this column here is all the strains combined, and this is a fully diverse from a hive bee microbiota. And here's microbiota free. So if you look at CFUs of this pathogen, Serratia marcescens is a strain taken from bees. It's a pathogen in bees. Basically, the microbiota free bees are very susceptible, and these die very quickly. Each one of these single bacteria, monoinoculations, actually offers some protection, but all of them together offer more protection, and the full gut homogenate offers more protection. So basically, there probably are a number of mechanisms at work. We looked in more detail at Snodgrassella, tried to figure out if it's acting through the immune system of the bee. That's one route. And so even if you give heat-killed Snodgrassella or heat-killed E. coli, you get some protection. That suggests the bacteria is doing nothing. It's just stimulating the innate immune system, the pathway that we expect gram-negative cell walls to stimulate. And it gives some protection, but the living Snodgrassella give much more protection. So there's something the living cells are doing. We're trying to chase down now what are they doing, but I'm not going to talk. We don't know yet. So, but we do know that they modulate the host immune system. This just shows that they stimulate host immune production of these antimicrobial peptides. But when you look at the signaling genes, so this is looking at RNA, basically transcripts, quantitative PCR of transcripts of immune system genes in the bee following these treatments. It turns out that everything stimulates the production of antimicrobial peptides compared to not having microbiota. But the living Snodgrassella somehow suppress some of these signaling genes in the IMD or toll pathways, so cactus, dorsal, relish, or some of the upstream signaling genes. So there's this network of crosstalk among these pathways, and it's doing something to actually modulate that. That's one thing we're interested in looking into, trying right now. One idea is that there's actually symbiont-produced RNA that's affecting the host. And from some other work, we know that RNA produced by Snodgrassella in particular can enter the bee. It can go all over in the bee. So we had we put in a plasmid for making some double-stranded RNA for GFP, just random sequence. And we can see that if we look in the gut of the bee, not the gut, the abdomen minus the gut, we expect it to be in the gut because the symbiont's there. You get this elevation, and even in the head of the bee, you can see this RNA being present. So it's somehow entering the bee, traveling around, and causing things to happen. And we originally got into this because we wanted to exploit the bee's RNA interference pathway to kind of do things in bees, and we thought we could use symbionts to deliver double-stranded RNA that would trigger it. And it turns out that worked amazingly well, largely due to these two people, Sean Leonard and Jeff Barrick, where we engineered Snodgrassella to deliver double-stranded RNA to the bee to do different things. And in that course of that, realized it's actually entering the bee somehow. So we are probably piggybacking on something that happens naturally, and we used it to achieve these certain things, one of which was stimulating the bee's RNAi pathway to attack this deformed wing virus, the major virus that's a problem in bees. And so we can see that, so up here are the ones that are not exposed to virus. Down here they're exposed to virus. And the ones that had the double-stranded RNA specifically targeting the virus have a survival advantage. So this is a bee survival curve. So, and we could show that it was triggering this pathway in bees, the expression of the RNAi pathway genes. And then we, even a little more surprising, we could target these mites, the other biggest, probably the biggest pest of bees is these varroa mites. They're actually vectors of the virus, but they also are pests in their own right. And so we made double-stranded RNA that targeted essential genes of the varroa mites, genes that were required for varroa mites to live. And varroa mites have their own RNAi pathway. And so basically we triggered that in order to kill the mites. So varroa mites are feeding on the hemolymph and fat bodies of the bees. They're ingesting the double-stranded RNA that's entered the bee, triggering their own RNAi pathway and causing themselves to die at a higher rate. And then just recently, Quang Hong in the lab did some similar work, very similar to this, with these microsporidians, nosema, that are big pests of bees, and they have their own RNAi pathway. So again, attacking their essential genes, you could use the symbionts. And the nice thing about this compared to just normal RNA, it persists in the bee. The symbiont is continuing to make it over time. Okay. So just finally, and I've never worked on these, so I'm not going to say anything really about them, but environmental microbes can also have a big impact on hosts. And even the microbes probably don't care. They haven't evolved to be in the hosts, even they live in different environments. But the hosts are very much affected by them. And this has definitely been shown to be the case in many different organisms. And examples are Drosophila and mosquitoes. A really striking example is that mosquito development requires this bacterial presence. This work by Carrie Kuhn and Mike Strand and others now has shown that they can't complete development without bacteria around. And so even though those bacteria can really be very diverse, different bacteria, they're not any one thing. So there's this role despite the fact that there's nothing that's really co-evolved there. Okay, so to summarize, what have we learned? First of all, the main message from this is really to understand insects, we have to consider symbionts. They are affecting all kinds of things, nutrient provisioning, resistance, thermal tolerance. There's examples of this where they detoxify insecticides or plant toxins, and they're used as cues in development and immunity development. And they differ, but they're different. They're not all the same. I talked first about these symbiotic rabbit hole, like Buchnera, the ones that are basically undergoing this decay. The hosts are dependent on them, but basically these symbionts are doing less and less for the host over evolutionary time. A major distinction here is whether the symbionts are still undergoing recombination. And so when they're not, they're just going to go down this clonal rabbit hole, just like the Y chromosome in mammals, where you lose more and more and are doing less and less. So it's a very similar process. But in other cases, such as Hamiltonella and also the bacteria in the guts of insects, like in honeybees, gene uptake by symbionts is really a source of new capabilities. So Gilliamella's acquired pectinase and all these things that break down the pollen coat, which bees can't do and couldn't do before. Surprisingly, this bacterial to insect gene transfer is coming up again and again, so it's interesting. We'll see how many cases and how much we can understand about that. And there's also applications of this. I just want to point out that there's a lot of efforts now to use the bacteria as either targets for control measures, or maybe in the case of bees as ways to prop up health. And so there's a lot of cases of that, and they're in human, like vectors of diseases of humans, animals, and plants, and crop pests, and pollinators. So insects are very important in human life, and symbionts are sort of one way we might be able to more easily control them. So unanswered questions. Well, I mentioned as going along, we really don't know how much of this works. We don't really know what molecules are being exchanged almost ever. We hope there are some general rules that not every case will be just totally different. Specifically, what signals are moving between partners. We're looking now into vesicles from Snodgrassella that are delivering things to the host. We think that is a likely route, but we don't know. There's several studies by others looking into sort of AMP-like peptides produced by hosts that are in bacteriocytes. And then we know that symbionts have to deal with the immune response of the host, so they're somehow integrating themselves and not being killed by the immune system. And just these are kind of extensions of that. What are the fundamental barriers to formation? There's whole large groups of insects, such as the Lepidoptera, the butterflies and moths. They don't have any intracellular symbionts. It's a huge group. And yet it's evolved again and again in beetles and in these hemipterans. So there's probably something basically different about their immune systems that's allowing it in some cases and not others, it seems, if you just take a bird's eye view of this. And then on the other end, they've really evolved just from a couple of bacterial phyla. Almost all the cases are from the proteobacteria, the Pseudomonadnota, whatever it's called now, and from the Bacteroidetes phyla. Almost all the cases are from those. And there's a lot of bacterial phyla that are in the environment but that are not participating in this. Okay, with that, it's 2:59. Sorry. I want to acknowledge people. I tried to show as I went along people who contributed to specific studies, but I've been super fortunate over the years to work with fantastic people. And these are all people who worked on endosymbionts in the lab, most of them gone now with their own labs, people working on the bee gut system, and some of them just really stars. And several of these have their own labs on the bee gut microbiota. And then major collaborators, so Paul Baumann and Linda Baumann early on got me into this. I'm very grateful forever for that. Molly Hunter at Arizona, Jeff Barrick and Andy Ellington, and Howard Achman at UT Austin. We've worked together all the time. So with that, I will thank you. And I hope there's time for a few questions.
If folks are free to use the mics. I do have a question from online that this seems terribly interesting to me. I'm over here. Hi, Nancy. Hi, over here. Hi, hey, okay. So this is coming from online. What are you learning? What you are learning about B vitamin relationships and heat sensitivity is fascinating. In humans, there are B vitamin issues and low heat tolerance in patients who have mitochondrial dysfunction and disease sometimes. Is there a connection between your findings on symbiosis and work that could be done to help patients with mitochondrial disease? I know it's a deep one, but.
It would be, yes, I don't know for sure. There could be. In the case of these symbionts, they actually have the biosynthetic pathways to make these molecules that are required for different enzymatic pathways. So how much, I really can't say if there's a connection. I don't want to speculate.
Sure,
sorry, but
please, up there.
Good question, though.
Okay, hi. Really enjoyed your talk. I had a question about the Hamiltonella. So they were carrying this phage, and you said without that selective pressure, they'll lose the phage. And I was wondering if any of these toxins had effects on the aphids, negative effects, and that could be why, because you mentioned that for the other example against the fungal pathogen.
Right. I don't know that it's been looked at extensively, but I think even the Hamiltonella with no phage are some metabolic burden on the aphid. So it's actually small. It's a small effect. Carrie Oliver did some experiments. You can't really, if you just grow aphids here, see how many babies they have, you don't even detect it. But if you compete them, in a selection experiment, so aphids with and without the Hamiltonella, and I think that's even with whether the phage is there or not. If anything, the phage actually keep the titers lower within the aphid, which actually makes it less of a burden for the aphid, because it's basically a metabolic, it's replicating in the hemolymph of the aphid. So I think the main thing is that it's a direct burden to have this extra thing in you if it's not doing you any good. So I didn't really go on about it, but there's these levels of selection. So at the host level, if the only thing that survives the parasitoids has Hamiltonella with a phage, then that's going to persist, and the phage are going to stay there. In the lab, we have Hamiltonella with the phage. It's killing the within host selection. It's killing itself. So it bursts, basically. They do lyse and deliver these toxins that way. And so, it's not in the interest of the individual Hamiltonella to lyse. So any mutant that didn't have the phage, that lost the phage would outcompete within the aphid. So there's sort of these levels of selection that you have to consider.
Yeah, it sounds like Buchnera has an extracellular phase. So is anything known about how the bacteria get in and out of the aphid cells?
Yeah, that would be great to know. It was worked out, actually, Ryuichi Koga, who's spent some time in our lab, but he works in Japan. He did some really detailed microscopy, and it wasn't known how it went from the maternal bacteriocyte to the embryo. And he determined that they are actually exocytosed by the bacteriocyte, which happens to be, it's always the bacteriocyte that's almost touching the embryo, right? Because they're in them. So somehow there's a signal between an embryo at that stage. It's a specific, stage 7 of the stage development. It's like a blastula stage. There's some kind of signal, and basically the membrane ruffles and it starts spitting them out, and they're briefly extracellular. We actually, after he figured that out, I realized, if that's how they're transmitted, then we could actually replace Buchnera by just flooding the body of the aphid with a new type, and then just looking for progeny that got the new one. And because it's actually extracellular, and that worked right away. We'd never done it, but actually it works pretty easily. So, but nothing is known about what the signals are. I had some suspects, but I'm not sure. There's a couple genes I'm really interested in, which are these only orphan genes in Buchnera. It turns out all Buchnera maintain them. Now we have many, many genomes of Buchnera. They go way back. They're super fast evolving. They're the fastest evolving protein coding genes in the genome. And I've always wondered what they're doing, somehow co-evolving with the host, I think. And they're secreted by the Buchnera has a flagellar basal body, no flagellum, but basically a type 3 secretion system. So I think some things are being secreted, and maybe those are communicating, but we don't know. Yeah.
Hi, great talk. I was just wondering if how do these insects regulate the burden of their symbionts? Is that known? Like in a population that is healthy, do you see a same level of symbiont like mosaics in the population? Is anything
Right,
known
so
about
for
that?
the endosymbionts,
Yeah.
yeah. Well, again, it's one of those things we don't really know. We know in aphids, they start off life like once at birth, they have a certain number of bacteriocytes, and those don't divide further, so there's got a set number. And then as the aphid ages, those bacteriocytes are killed off, and the mother loses them over time. And this lab in France, that Simonet et al. Paper, PNAS 2018, I think, did this beautiful work, like just imaging that, there's this very kind of non-conventional apoptosis pathway to kill off these bacteriocytes. But how that works and why, what communication. There's clearly something, because you can at some stages, all the Buchnera are dividing. You can see them dividing in the whole bacteriocyte. The bacteriocyte next door might have none of them dividing, so there's some kind of signal within that cell, but we don't know what it is. Hopefully someone's going to figure this out soon. There's always new methods that offer hope for getting past this fact that we can't culture them.
Yeah.
Sorry.
I really enjoyed your talk. I just kind of had a curiosity question about the bees and the symbiotes that help them. And so I was wondering, is it during the pupa stage that they get inoculated normally with?
It's after they emerge from the pupal stage. So they're adults. They're, the youngest adults, newly emerged adults.
Okay.
So the pupal stage is when they're still in there, they're not eating. And then they emerge from the pupal stage in the hive. They would chew their way out. And immediately, the nurses are actually helping them get out. And they're immediately exposed, and they start to acquire the symbionts. But yeah, so if you prevent that, you can have them not have any symbionts.
Yeah. So then once the bee's an adult, does the age matter? Does it matter if it gets inoculated right away, or if you wait and then were to inoculate later, does it affect the bee's ability to form symbiosis with these organisms and then be resistant to certain pathogens?
Yeah, we haven't actually done exactly that. That's interesting, yeah. Is there like a open door at a certain stage? And we haven't done that. We generally inoculate them within 1 day of emerging, and it goes to full populations of all of them by day 4. But if you prevented it and then later expose them to the true, the normal symbionts, we haven't actually done that. I don't think anyone's done that. It's interesting because it could be. The gut itself does mature, so it's an adult, and so insect, development, it still goes on in the adult stage. The gut is still maturing. The bees are gaining weight by eating a lot. There's a lot of stuff happening before it becomes a fully, a really fully mature adult bee. In the older bees, you start to get a little bit more disruption of the microbiota with kind of pathogen-like things being a little more common. They're still a minority, but the older bees start sort of like in older people. The microbiome is a little bit more erratic with environmental microbes being present, although it's still dominated by these main symbionts.
Thank you.
We should retire to the reception and invite everybody to join us and
Okay.
discuss.
Yeah, no, I was going to say the CME code. Oh, good, 50096 for the people listening at home.